I'm Bill Dantzler, Professor Emeritus of Physiology at the University of Arizona, and today I have the privilege and the pleasure of interviewing Dr. Douglas G. Stuart, Regents Professor Emeritus of Physiology, also at the University of Arizona, for the Living History Project of the American Physiological Society. Now, for over half a century, Doug has been a leading figure in the study of spinal cord neurophysiology, especially as it relates to posture and locomotion. He has pioneered a bringing together of groups of people, both nationally and internationally, to further these studies. At the University of Arizona, he has been the moving figure behind the establishment of a number of campus-wide scientific programs, the most notable of which is the outstanding campus-wide program in neuroscience. For these many achievements, Doug has not only been appointed a Regents Professor by the university, but has been awarded an honorary Doctor of Science degree. Today, we're going to explore with him some of his experiences that have led to his accomplishments and some of those accomplishments. Now, Doug, it's a real pleasure to have you here, and I'm delighted that you're willing to take part in this interview. As people who don't know you will recognize as soon as you start talking, you were born and raised in Australia. Were there any aspects of those early years, or your grade school or high school years, which you think may have, in retrospect, predisposed you to going into science, or even possibly into physiology?
Well, Bill, thank you for this opportunity to participate in the Living History Project of our society. I like this, and I particularly like the idea that the younger members of our society will look at these and see whether there's anything we much older folk did that is relevant to what they're doing today. Now, as for starting out in grade and high school, I grew up in this very attractive town halfway up the east coast of Australia, just 20 miles in from the ocean, 500 miles north of Sydney, and I can assure you that in my day, males did not take biology courses. They were for females. However, that was the culture. However, I had very good math instructors, and in calculus, for example, I was doing differential equations when I was 14. And in addition, I had really good teachers in English, chemistry, physics, and that helped later on.
Well, were there any particular teachers in grade school or high school that influenced you particularly?
In retrospect, the teachers were very conscientious about teaching, but they weren't into counseling, nor were there any official counselors in the state of New South Wales in Australia when I was in high school.
Okay, well, you began your initial higher education by getting a diploma in physical education and then teaching in high school for 3 years. Well, what led you to go in that direction? Why did you decide to do that?
At the beginning of World War II, my state began a national fitness camp program. Now, these camps were attractive, quaint in a way, wooden structures, always near the ocean. And the idea was that if you were in grade school or high school, college, if you were in the private sector, young in the private sector, older in the private sector, you could go to these camps for long weekends or for a week. A lot of the time, an attempt was made to mix the people up. The purpose was national fitness, physical and mental fitness. There was absolutely no politics in this. It was fitness. And you would go to the camp, you would have formal exercise training, very relaxed lectures about physical and mental fitness. You would swim, you would hike, you would interact with the other people. And then at night time we would sing Australian folk songs, and we would dance with the movements of the dancers tied to the Australian culture and folklore.
I think we'll forego the singing and dancing for this interview.
So anyway, I got involved in this when I was about 14 years of age. When I was 16, just at the end of my 5 years of high school for university entrance, I became a junior instructor. I really liked the people who ran this program, and they said if I got a diploma in physical education, I could stay close to the program and maybe work for it one day. So that's how that came to happen in my life.
Well, did this, what sort of influence did this period then have, and that you're getting your diploma and teaching in high school, on your later career?
Well, to begin with, you have to realize I'd barely turned 16 when I began at Sydney Teachers College in Sydney. Now, there were 3 classes, you see, a 3-year diploma, 3 classes of people doing this program, 20 males and 20 females in each class. Now, among the males, over 80% of them were ex-servicemen, because I began in 1948. And I got to know these people, and I heard many a story about the campaigns in North Africa, Europe, Asia, World War II campaigns, terrible stories that really had a profound effect on a 16-year-old. Now, the college program, I still remember the teaching of English. I remember a wonderful educational theory teacher, and I greatly respected that. I enjoyed those years. I acted in plays while I was at Sydney Teachers College as well. And then I went out and became a high school teacher, and I taught in quite a prominent agricultural high school on the outskirts of Sydney. And that was a very interesting experience, because among the 500 students, 250 of them lived there. They lived on the grounds of the high school, boarding school students. And I had a lot of experience in getting that group together, groups of them together, to do certain things. That was very good, I thought. And I enjoyed my time talking to the parents of the boarding school teachers. You must remember that I was 19 then and teaching students who were 12 to 18. So this was a very unusual and a very interesting experience. But after 3 years of it, I realized that the time had come for me to stop teaching and to focus on the physiology of exercise science, go back to school.
Okay, so you felt that you needed more education, but you decided to leave Australia in 1954 and come to America to study at Michigan State University. What led you to making that decision for the continuation of your studies?
If I was going to have a career in Australia in the field of physiology of exercise, national fitness, that type of thing, I would need a bachelor's and a master's degree in exercise science or their equivalent. Well, no such degrees existed in Australia at that time, but they certainly did in the USA. And I'd like to emphasize at this stage that I'd read a ton of US literature by then. I loved American literature. I began that in early high school days. I continued in college days, and I did it as a young high school teacher. So there was a lot bringing me to the USA.
While you were at Michigan State, you changed direction somewhat. You decided to go more in a complete direction of physiology, and particularly neurophysiology. What led to those changes?
When I arrived at Michigan State, I knew of the field of the physiology of exercise. I didn't know that much about it, but I knew about it to some extent. And what happened to me there was that I really fell in love with the field of physiology. I really liked the lectures, and I sure liked the laboratories. Now, as you know, we used to, the laboratory results, we used to put them out on a chymograph. smoke chymograph. That was great fun. So that happened to me, and so what happened, I not only became interested in physiology, but I expanded my interest in physiology to cardiopulmonary physiology and to neurophysiology. I had a wonderful professor of physiology. He had a PhD from Princeton, Dwayne Collings. He was my primary physiology mentor. He wanted me to go to UCLA and do a PhD in physiology because he liked the faculty there. And in addition, there was a young physiologist, a beginning assistant professor, Herman Rudenberg, who had just completed a PhD at the University of Chicago, and his first course at Michigan State was a laboratory course in neuroscience. That was a totally new thing for me. New thing. Neuroscience was new for me, and it had a very powerful effect on me.
So in response to your teachers, you decided that it would be a good idea to go on for your PhD. So you did go to UCLA to get your PhD, and then you did some postdoctoral studies as well. Do you want to tell us a little bit about your teachers at UCLA for your predoctoral studies and your postdoctoral studies?
They were an extraordinary group of people. My professor, Alan Hemingway, was a physicist turned physiologist. He was just a tower of strength and support for me while I was doing my PhD. And he had in his laboratory a postdoctoral fellow, trainee, who was a fourth generation Yale-trained clinician. He may even be unique in the USA, for all I know. But anyway, Walter was marvelous with me. He was very stern, very strict, and he taught me that one had to work awfully hard, focus awfully hard in order to make just a small contribution in the field of physiology. In retrospect, that was a very important thing. But then again, there were many other professors. While I was a PhD student, 4 of my professors became members of the National Academy of Sciences. Ted Bullock in invertebrate neurobiology, Ted Magoon in sleep-waking mechanisms, Don Lindsley in physiological psychology, and actually neurophysiological psychology, and Tom Sawyer in neuroendocrinology. They were all great scientists. They were all incredibly engaging and supportive of the students. So that was a very powerful effect on me. And then as a postdoc, first as a 2-year postdoc, and then as a research assistant professor, I worked for 4 years with Earl Eldred, who was a professor of anatomy and very well known on the properties of muscle receptors and what role they might play in the control of movement. Earl and I wrote quite a few papers together in those four years, and we had some terrible fights about how to interpret the literature, how to write the papers, but we were very, very close friends. So we enjoyed those battles, and we're still friends all these years later.
Great. Well, as you know, collaborations are very important in science, and they're becoming even more important these days in physiology. And no one has been more collaborative than you. So would you please tell us a little bit about some of the collaborations you established early on and later on in your years, and how did they develop?
Yeah, well, I'd like to talk about this, particularly with the younger members of our society in mind. Clearly in a collaboration, the parties collaborating have to feel they're really getting something out of the collaboration. But there are additional issues. I believe that research should be fun, F-U-N. And I believe that if you collaborate with somebody, you should really enjoy them as human beings. You should enjoy their company. You should enjoy what they've done and how you interact with them. So I feel very strongly about that type of thing. Now, collaborations can be planned, or luck and chance can come into play. So I'll give a few examples. First and foremost for collaboration was a research engineer electronics expert who later became a computer expert, Bob Reinken. Bob Reinken joined me at the University of California, Davis campus, in 1965, and he worked with me until 2000. 35 years of work together. What did he give me? He gave me this very sophisticated electrophysiological lab. I had a pretty good lab when I went up there from UCLA, but he really improved it. And there's a photo of Bob and me here for this program. The thing that he gave us in addition to the laboratory was that he was very good about the systematic organization of data. That was very, very valuable. Everybody that came to my lab had that opportunity. And when I think of Bob, I think of, for 35 years, I also think of Pat Pierce, who worked with me for 27 years. She'd been a high school biology and English teacher, and then when she had the opportunity to come and work in the lab, she was very, very happy about this because she really liked the research environment at the university. Now, with that background in biology and English, she became a really good technical editor. Oh, she did the traditional biological work, and the photo shows her peering at a microscope, but she was an amazing editor, and that proved to be very, very valuable. Valuable for 27 years. Now, I believe that people like Bob and Pat, their value in experimental work, you cannot overestimate it. So that was planned, obviously, to have them. Now, the second example I want to give is pure luck. While I was a PhD student at UCLA, I had the good fortune to work for a year with Yajiro Kawamura, who, a Japanese professor. He, at that time, was the youngest professor and head of a physiology department in Japan. He was a former neurosurgeon and 38 years old when he got to UCLA in 1960. And Kawamura had come on a very prestigious Rockefeller grant, but there was no lab for him. All the labs were full up, so here he was working with me. So I had the luxury, the luck and the luxury to work with him for a full year. He too was very good on the systematic thinking about each day's experiment. He was very good for me in that way. I learned a great deal from him. And in return, I helped him with his English, to give talks, to write papers. I even taught him to drive a car. In fact, one Sunday morning on the UCLA campus in a huge parking lot, there was only one other car and he happened to hit it. So we had these wonderful experiences. Now we are still, over 40 years later, very close friends, and there's a photo of Yajiro for this interview. And so that was an example of luck. Now the next example, and again I'm talking to the younger people in the society, the next one was where the planning is, as it were, from on high. I had arranged when I was at UC Davis to have a Nobel laureate from Australia, John Eccles, come and give a week of lectures. And he came, did a great job, and talked to many people. Now in my lab, he thought that we had good electronics, but he didn't think I personally knew enough about the nuances of doing work on the mammalian spinal cord. I didn't know all the tricks of the trade. They'd been developed in the 19th century, in the late 19th century, particularly by his own professor, Charles Sherrington, who was also a Nobel laureate. And then Eccles, later on in Australia, had advanced these techniques. He wanted me to work with Anders Lindbergh in Göteborg, Sweden. At that time, we're talking about 1966, Lindbergh was considered the world's leading spinal cord neurophysiologist. I couldn't go that year. But in 1971-72, I was able to go for 8 months. Now, by then, Lindbergh, and I have a photo of him lecturing, a very intense man, lecturing like this. He had acquired as a permanent member of the Department of Physiology at the University of Göteborg, Elżbieta Jankowska from Warsaw, Poland. Her ability in spinal cord neurophysiology was and still is absolutely amazing. So here I was in Göteborg working with these extraordinary people. I call them my latter-day mentors because I was a professor by the time I worked with them. And Lindbergh, because I was a professor, took me, we hit it off very, very well, and he would talk to me about issues in his life as a professor, and he did that up to 2 days before he died in 2009. And we were very close, talked a lot about issues, and then while I was there, he met me every Saturday morning for several hours to talk about the history of work on the spinal cord and the strategy of research. That was an extraordinary experience. That was remarkably generous of him to do that. So that was an example. And now let me give you, the younger people, an example of chance in the collaboration. I came out of the end of an international meeting in Paris. I was walking back to my hotel when I saw Tony Taylor and John Stevens sitting at a table outside a bistro. Now, Tony Taylor was the head, at that time, was the head of physiology at the Sherrington School of Physiology at St. Thomas's Hospital Medical School in London. Tony and I did not know each other, but we knew of each other's research. We had both worked on the biomechanics and the neurobiology of tremor. Why did different types of tremor occur at a certain rate? But we had never met. So we began chatting. Stevens, the graduate student, had just given quite an impressive talk at this particular meeting. So what we did, we met, I introduced myself. We walked around Paris that night till long past midnight. I would have met them about 4 p.m. We must have talked till about 1 a.m. And by then we'd agreed that Stevens should come to Tucson and have a postdoctoral experience, which he did, and that Taylor should come and we should do some collaborative research. And that too happened. And that was a very, very, very rewarding, enjoyable collaboration. These were British physiologists. They had a strong feeling about physiology in the UK. And we used to like to compare what was going on in the UK versus the USA. We had a wonderful collaboration. So that was very, very good, and that was an example of chance. Now I'd like to move to when you're running your own lab, and let's start when you're young, you're beginning. You're a research assistant professor, you're not tenured, you're just getting going. Well, I think when that happens to you, you should look around and see what other people are in the same boat as you. And I met Jim Hayward, who had my position, not in physiology, but in anatomy. His research area was neuroendocrinology. But Jim was a board-certified neurologist, trained in a premier program at Harvard, and very curious about some of the things I was doing. I found him very, very interesting. I learned a great deal about neurology, and I learned a lot about primate neurobiology from Jim. And in return, I taught him how to record from extracellularly from cells in the hypothalamus, which he later did in his own laboratory. So that's an example of two young people starting out in their research programs and working together. And in addition, as I've emphasized a few minutes ago, we greatly enjoyed each other's company. So that's when you're young. Now, what about when you're a bit further along? What about when you're getting up there, you've got tenure, you're an associate professor? What about collaboration then? Well, in my case, when I went to UC Davis, in 1965, I met a young graduate student in zoology who was working under a premier zoologist, Milton Hildebrand, an extremely good zoologist. This was a chap named Ted Goslow, and he came to my office a couple of days after I arrived there for some advice on an experiment. So we got to know each other. There's a bit of chance here. I came down to Tucson 2 years later, and he went to Northern Arizona University, about 280 miles north of Tucson. Now, I suggested to Ted that we should collaborate. He was an expert on comparative vertebrate morphology. My expertise was in neurophysiology. So, he taught me a lot about comparative vertebrate morphology. I taught him a lot of neurophysiology, and we actually found a way to combine the two disciplines, to combine them. And we wrote articles together for over 10 years. So that was a collaboration between universities. What about in your own university? Well, in my case, in our Department of Physiology, I was very fortunate to, when Zia Hassan took a position in our department as an assistant professor. That was about 1980. Zia was formerly an electrical engineer turned neurophysiologist, and he and I began working together, and we had a very productive association for 10 years. Not just in research. We tended to write conceptual papers together, but I learned a great deal from Zia, and in return, I helped him with his career. And at the same time, well, actually a year later, another assistant professor arrived, Roger Anoka. He arrived in the Department of Exercise Science, and we later gave him an appointment in physiology. Roger's expertise was in biomechanics and kinesiology. Well, I'd had a lot of this, but I needed more. And Roger was a great teacher. And in return, I got Roger into intracellular recording in the nervous system, etc., etc. Now, Roger, in the case of Roger and me, our first published paper was in 1983, and our most recent one is in 2011. So that has been a very productive association. Now, finally, I have something that I want to really stress, and that is what is your relationship with your own trainees? So in your lab, you've got research undergraduates, medical students, predoctoral students, master's degree students, PhD students, postdoctoral trainees. I believe that certainly you are their supervisor, but they're your collaborators. They're your collaborators. You work with them. And the atmosphere in the laboratory should be one of collaboration. Their attitude about you should be that you are their collaborator. Now, in my case, quite a few of these people, I felt that they were much better trained and would go much further in neuroscience than would I. And I put a lot of effort into the strategy of not only their research, but of their academic careers. Now today, many of these people are professors in the USA and abroad. They're at the top of their fields. Now in the USA, these people are at the universities of Arizona, Colorado, they're at Washington, University of Washington, Washington State University, the Barrow Neurological Institute, the Mayo Clinic Medical School. They're at leading places and they're doing leading work. And in the USA, what about abroad? Well, these people are at McGill University and the University of British Columbia in Canada. They're at University College London in the UK. There's a chap in the Institute of Physics at the Bulgarian Academy of Sciences. There are people at the University of Newcastle in Australia. And there are people at two Japanese universities, Nara and Ibaraki, and their programs are very tied to programs at the universities of Tokyo and Osaka. So there are quite a few distinguished universities, and two of these former trainees, these former collaborators, have their own institutes. One's at the University of Chicago, and one's a freestanding one in Nicosia, Cyprus. And I would like the young people to think about this. These people who have done so well were once in my laboratory, but they were there as collaborators. They were there as collaborators.
That's extremely interesting, Doug. What do you think are some of the most significant contributions that resulted from these collaborations?
Well, in my case, you know, for 50 years or so, I've been working on the control of movement with a particular emphasis on the role of the spinal cord. Now, the type of people who do this work talk about it as either being inside out or outside in. Inside out is where you're recording either extracellularly or intracellularly from single cells, in my case in the spinal cord. You're recording from the afferent impulses of single mechanoreceptors. You're recording from the ventral root filaments of single motor neurons, and you're studying the neuromuscular properties of the apparatus that is controlled by the brain. So that's all inside out. And you know, those experiments are very long. Those experiments, when you're working on the cat spinal cord, surgery for 9 hours, and then if it's successful, you're going to go all night. I did that for 30 years, all night experiments. And what do you do with the information? Well, the information you're generating inside, you infer what is it doing on the outside? What is it doing about the movement itself? So that's inside out. I did a lot of that. Then what about outside in? Well, outside in is where you analyze the actual movements of the animal, and then you infer what rules must you could say rules, you could say responsibilities, does the nervous system have to produce those particular movements, outside in. So in my case, a lot of my outside in work was on the footfall patterns of cats converting from low to high-speed locomotion. We got a great deal out of that type of work. About inferences about what reflexes must be doing in order to produce these particular movements. So this was sort of how all the time went for many, many decades. But there's something I also did, and I started this actually as a completing graduate student. I decided that if I was going to move into a field, I would write a conceptual review about it before I actually generated data. I would write a review, short or long, about what I thought was going to happen once the experiments began. And I first published a major article on that in 1961, just 3 months after I completed my PhD. Now, I wrote traditional reviews. In the 1970s, Mary Wetzel, a physiological psychologist here at our university, and I wrote a very long review on the neural control of cat locomotion. In that particular review, we introduced a lot of people in the West to the work of a group of scientists in Moscow, USSR at the time, and opened their eyes about some amazing work going on there. So that review, because of that alone, has been used in PhD and postdoctoral training programs for many years. Now, in the 80s, Zia Hassan and I wrote an outside-in review about, well, just what do all these proprioceptors do? What is the role of proprioceptors, muscle spindles, tendon organs, and so forth, in the control of movement? So outside in, we discussed the movements of the crab, the cat, and the human, and we inferred what might be happening. that is still under consideration, even though that review came out in the 80s. Now then in the 90s, Roger Anoka and I wrote a different kind of review. By then we were working on the neurobiology of muscle fatigue, and we proposed, about 1994 or so, 4 themes of work on the neurobiology of fatigue that we thought should guide research, not only in our own lab, but in other labs. And that's exactly what happened. For about 10 years, those ideas were tested in several laboratories. And then, now we're getting into the 21st century, in 2004, Ed Stouffer, who was my first PhD student, graduating in 1974, 30 years later, he wrote a review with me on a totally different level of analysis, and this was at the very cellular level of thinking about intracellular recording, on challenging conventional thought on the role of after hyperpolarization in the firing pattern of cells. So I've always believed that, as well as the experimental work, conceptualization of what might be happening is an important issue.
Well, in addition to that, you, and with your many collaborations, you also were able to bring together groups of scientists, both nationally and internationally, for symposia. And some of these resulted in symposia publications. Do you want to tell us about those symposia that you were able to establish and the sorts of publications that came from them?
I like the way you express that, Bill, that the symposium should occur, and then there should be a symposium volume or a sequence of articles in a relevant journal. Well, I strongly believe in this, and I'll talk about a couple of examples, different types of things. One that was involved with and still am, but to a lesser degree, is on the neural control of locomotion. Now, we have this particular symposium every 10 years. We wait 10 years to see what has occurred over the 10-year period. So we gather together a group of people, we discuss the neural control of locomotion, we publish first a book and now more recently articles in a sequence of articles in a journal on the neural control of locomotion. Who are these people? Well, by design, these people study either invertebrates, non-mammalian vertebrates, mammalian four-legged animals, non-human primates, and humans. We get the whole shebang from the most primitive vertebrate to the human in a phylogenetic sense to discuss and what's going on in locomotion. Now, the reason we do that is that we believe that philosophically, if you like, or strategically, if you think about the control of movement over the entire animal kingdom, principles will emerge on the control of movement. And to this point, that has continued to occur. If you do this, to get all these people together, obviously they've got to want to come and discuss these issues. The organizing committee has to be one that has the respect of the entire field. And you have to know, as did I, when to move out of being on the organizing committee and have a younger person come on who is very, very active in the field. My work changed somewhat in the late 90s, so I retired, as it were, as an organizer. Another person came on, and that's going to keep going on. I think this particular sequence will go on for many more years and contribute. That's one example of a symposium group. Now, there's another type of symposium that meets a societal need. And here I'm talking about the Bulgarian Academy of Sciences that ran an international meeting on the control of movement. And they ran those meetings every 3 or 4 years from 1969 to 2004. These meetings were held largely on the Black Sea, those lovely beach cities that on the Bulgarian coastline. And the purpose was to bring together people from the Eastern Bloc, as we called it, Eastern European countries, Soviet Union, later after 1989-90, Russia and so forth. And the idea was that we wanted these people to have a really good relationship with their colleagues in the West. And I worked quite hard on this particular group of Symposia. I started on the executive committee about 1981, and I want to emphasize just one aspect of it. For the 1993 meeting, I brought back to Tucson, or had brought to Tucson, chapters by everybody who had participated in the meeting. That was 94 chapters, 63 of which were written by people in the Eastern Bloc. So what we did with them, we edited them for their English, and had a lot of interaction with them as we did that. And then I actually had that published right in Tucson, actually published the whole shebang. And there, Pat Pierce was a great help, but I also had the help of local colleagues and colleagues throughout the USA. Now, the end result was a book, a symposium volume, where their work was presented in acceptable English. English that they could use as a model in their own laboratories when they wanted to talk to people in the West again, or when they wanted to write papers and submit them to English language journals. Many of those people have told me that they used that model for many years. And I believe very strongly that for those of us whose primary language is English, we have to help the people in our field whose primary language is not English. And this is what I did in that particular case. I've also done that in Japan. I've worked on had a symposium volume like that for Japanese work. And indeed, I myself have been editing Japanese papers from 1960 until the present day. I feel very, very strongly that we have to do that for our colleagues.
Well, let me skip back a little bit in terms of your academic career. In 1967, after 2 years as an associate professor at the University of California, Davis, you came here to the University of Arizona as a founding member of the brand new Department of Physiology in a brand new College of Medicine. Do you want to tell us a bit about what it was like playing a role in that sort of founding and in a new department in a new college?
Well, I think the first point is that we had a very good beginning department head, Paul Johnson. I think Paul's personal behavior in the areas of teaching, research, and service were a wonderful model for other physiologists on what they should be doing, thinking about what their philosophy should be as academicians, and to have a first-rate department. I give Paul a lot of credit for setting that example. I enjoyed the camaraderie of the initial people, particularly you, Bob Gore, Rafi Gruner, George Hedge, all people who were truly committed to doing a really good job, and we had a great deal of fun while we did it. Now that first year was rather tumultuous. I arrived in mid-August with Bob Reinken, and we immediately began getting ready laboratory exercises for the spring of 1968. We not only developed exercises that could be done, we did new exercises. We had our medical students do intracellular recording from motor neurons. Not motor neurons, I'm sorry. Neurons in an invertebrate in the abdominal ganglia of Aplysia californica. But this was, to my knowledge, the first medical school in the world that ever tried having the medical students do intracellular recording. But my goodness, it was a lot of work to get all that done that particular fall. However, we did it, and then we went on, and I think the department has flourished, not due just to Paul and me and you, but to people who came along.
Would you do
but I have others that I want to talk about too, because we not only enjoyed each other's company in our department, but we all made contacts with people, our pioneering faculty in other departments. Now, I was very close to the head of neurology and a professor of neuroanatomy. And the three of us, in the spring of 1968, we put on an interdisciplinary course in neuroscience. Now, the neurologist had come from Case Western Reserve, the anatomist from Harvard, and I'd had this experience at UCLA. But we put on an interdisciplinary course in 1968, and there weren't many of them in 1968. I'm kind of proud of that.
Would you do anything different today if you were, again, helping to start a new department of physiology in a new college of medicine?
Well, yeah. Well, obviously, you can imagine getting intracellular recording going in those days. We put a lot of effort into it. Why? Well, I've always believed that not just the science of physiology, but the teaching of physiology requires experimentation. And I think that physiology courses should be accompanied by laboratory exercises. The field has moved on, and I think that what we should be doing now in my area is putting, and possibly in other areas as well, I haven't thought as much about them, but we should be putting on virtual computer experiments. Our students should be, just as I was doing intracellular recording with them many years ago, and they were actually doing it, now they'd be doing that on a computer. They would be inserting microelectrodes into cells, they would be studying ion channels, etc., in a virtual situation. And not only in experimental animals, but there are some very sophisticated things you can do with humans too in a virtual format. I first saw this at the University of Western Australia in Perth in the about 1991. A young assistant professor showed me how he was impaling muscle fibers with a microelectrode, changing the extracellular environment, etc. And it looked tremendously rewarding, promising, and that's all been borne out. So I would be very keen on keeping that going.
Well, let's continue with some more things about your academic career in the university. One of your hallmarks of your career has been university service. And in that regard, you essentially pioneered and essentially were the catalyst for the development of a number of university-wide programs here at the University of Arizona. I think the most notable of which was the campus-wide program in neuroscience. And for these activities, among others, the university has awarded you an honorary Doctor of Science degree. Do you want to tell us a little bit about how you, as a single individual, managed to lead or take the lead in developing these campus-wide programs? What did it require? How did you go about it?
Yes, well, I've got to be careful not to be long-winded here because I have very strong feelings about this. I became very interested in this sort of thing at UCLA with Ted Magoon, who had an interdisciplinary bent. And then at Davis, I had participated in starting to get a new campus-wide program in physiology and bioengineering. Here at the UA, University of Arizona, that honorary degree was both for neuroscience and for biomedical engineering. And it's worth talking about both of them because we use slightly different tactics, but with the same principle in mind. And that principle is that if you're going to be introducing or trying to introduce a new program at a major research university, what you're trying to do has to not be self-serving, it has to be totally transparent, and it has to be totally academic. Now, in the case of neuroscience, it happened this way. The vice president for research and dean of the graduate school took me to lunch one day and asked me if I could suggest a new program of biology for the main campus, not the medical school, the main campus, that he would champion and help produce. Well, I had one in mind. I immediately mentioned invertebrate neurobiology, and then at the lunch I told him about it, and he said, okay, come back when you've got something for me. So I left. Within a day or two, I convened all of our neuroscientists at the University of Arizona. At that stage, they were all in the medical school. I think there were only 3 on the main campus. That was in 1980. So I talked to them, a remarkable group of people. They agreed that this was a good idea. They understood it wasn't for the medical school, it was for the main campus. And they told me to set up a strategy. So the strategy was to form an ad hoc committee on interdisciplinary neuroscience, a self-appointed ad hoc committee. And 5 of us from across the campus. So what we did is we wrote a position paper on the field of invertebrate neurobiology. And we got a bit of outside support for that, which I won't have time to tell you about, from colleagues who became interested in what we were trying to do. And in the position paper, we emphasized that we didn't just want our administration to listen to us or read what we said. We wanted an external consultant to evaluate that position paper. And we suggested a chap who later on actually won a Nobel Prize for his work using invertebrates in the study of learning and memory. That was Eric Kandel. Well, I took the position paper to the vice president for research. He read it, he liked it. He gave it to the president with his very strong recommendation. That VP was a physical chemist, and the president was a physical chemist. The president read the paper, and the first thing he did was invite the external consultant to come to the U of A to talk about this position paper and the field. That happened, and the president was greatly impressed with that external consultant who supported the paper very strongly. And so then the president changed our self-appointed ad hoc committee to the University of Arizona Committee on Neuroscience. That was the beginning of campus-wide neuroscience at the University of Arizona. And the president also gave us permission to suggest a budget for a person who would head such a group. And we submitted a budget, and he approved it. It was approved up the line in the state university system, and lo and behold, that's how we got our new head of invertebrate neurobiology, who showed up on the campus in December 1985 and did a great job subsequently. Now that's one way of being transparent, one tactic. When we go to biomedical engineering, and remember I'm using the word biomedical engineering because at our university agricultural engineering is a very strong field. But in biomedical engineering a different tactic was used, but the same principle was applied. Now in this case, by then I'd become the associate dean of research in the medical school, and I was interacting very closely with a new vice president of research and dean of the graduate school. Now this particular vice president, a biochemist, a protein biochemist, he was very interested in interdisciplinary programs. He wanted to help develop what later became known as graduate interdisciplinary programs, GIDPs. And I helped him with 2 or 3 of them. And then he said to me, well, Doug, what about biomedical engineering? He said, I don't know anything about it, but what do you think we should be doing? So what I then did is, with his financial support and wonderful support from the Dean of Medicine, over a period over a year, we put on 11 statewide workshops in different facets, different subdivisions of biomedical engineering. The people attending these workshops were from our own university, Arizona State University, Northern Arizona University, and from quite a few private sector businesses in this field throughout Arizona. So we didn't just put on the workshops. That VP and I met the night before each workshop with selected people who would be talking the next day to learn more about their sub-discipline within biomedical engineering. So we listened to them, all 11 of them, and then that vice president and I had long talks, just the two of us. What had we learned? What looked good? What should be done here? Well, this had a big effect on this vice president, and he became extremely interested in this now, after all of this, what I consider to have been totally academic activity. And he took it on himself to begin a graduate interdisciplinary program in biomedical engineering. And I want to add something here that I still get intense enjoyment from what I'm about to say. So here we have a graduate interdisciplinary program in biomedical engineering. But a new dean of engineering then, a few years later, started an undergraduate program in biomedical engineering, not to take the place of the GIDP, but to supplement it. And that particular dean had been a participant in one of those workshops that had been held back in 1994. So that was a good feeling. So in summary, be academic, be transparent, don't be self-serving, think of the university, and also have a very smart vice president of research and have a very smart president.
Well, that's very good advice. So finally, what advice do you think you would give to young people either considering a career in physiology or actually beginning a career in physiology? What advice would you give them today?
Well, I would give them the advice I have given my own children, my own grandchildren, and every person I come in contact with. Follow your own star. Follow your own star. Now, the star may change. There may be several stars in a lifetime, but always be following a star. And don't worry about the financial reward. I think that one should work to live. Work to live. Homo sapiens is homo faber. Actually, I've got that back to front. Live to work. Live to work. Don't work to live. The work itself should be the driving force. And if you want to join a physiology department or at a major medical school, you're going to have to wait longer because the faculty aren't retiring that rapidly, at least in the USA. I don't like that. I don't like that. I think that we should have a fixed retirement year. I'd make it about 70. It used to be 65 years ago, maybe. But now people are living longer and healthier. And we have many an example in the USA of faculty, older faculty, retired faculty who are still getting their extramural research support. Normally they're in emeritus status as you and I, and but they're still getting support if their work is considered favorably by the granting agencies. At our particular university, I think the record is somebody who was 91 or 92, Speed Marvel, who was a chemist. So I think that if we could solve that problem, that would help get the younger people jobs at a younger time, maybe after 2, 3, or 4 years of postdoctoral work, whereas right now it's much maybe 6, 8 years of postdoctoral work. So that would be another change I'd like to see. I am very worried about the cost of setting up the new laboratories for young people. That's going up, and research universities have to find ways of finding the money to do that. We have one person right now at the University of Arizona who is going to need a piece of equipment that costs $1 million. You didn't get that bill when you started at the University of Arizona. But you know, we have to do these things because this is where the science is. And where the science is, then the instrumentation has to be for the science. I consider this to be a very serious problem. I'd like to see the society thinking about this kind of problem too. I'd like to see all of us thinking about this problem. We have to solve this problem.
Well, thank you very much, Doug. It's been great to interview you. I think you've given us a lot of things to think about, and I hope the younger people who watch this will take advantage of those ideas. So thank you again very much for agreeing to this interview.